previous lecture, we reviewed retrieval, graphical rendering, analysis and prediction of structures of biomolecules, primarily proteins. Of course, we just scratched the surface of the explosively developing field of structural biology, which is an integral part of informational biology. In the next parts, we are going to add some basic methods of comparison of nucleotide and amino acid sequences across related proteins. These methods and associated skills might significantly enhance your own research capabilities. Together with the structural analysis, these methods will form a foundation which we can use to approach the challenging question of the origin of the SARS-CoV-2 coronavirus.
What if I tell you that the coronavirus behind COVID-19 pandemics might have been tweaked by humans, maybe just a little bit, but crucial bit, to conquer the world? What are implications for the biological future of humans if it were discovered that SARS-CoV-2 was a so-called gain-of-function mutant designed in a lab? Would it make any difference if we established that the virus had crossed to humans from a natural animal reservoir in a zoonotic event as many other viral epidemics before? Is this virus natural or not natural? Why shall we be concerned at all? After all, here is 2020 paper in Nature stating that COVID-19 is most likely of a zoonotic origin. In a nutshell, the answer is simple. If it comes from nature, it can be stopped through wildlife protection and conservation. However, if this one comes from a lab, we can be assured that the next and nastier and more deadly creation is imminent. In this lecture, we are going to explore the origin of SARS-CoV-2 using methods of phylogenetic analysis. Our goal is to figure out how we can argue for and against natural or artificial origin of COVID-19 using biodiversity and relatedness of the coronaviruses. This requires understanding of the basics of phylogenetic analysis. Clearly, those skills of reconstructing and making inferences from the phylogenetic trees can be directly applied to a wide variety of problems ranging from immunology, structural biology to sociology and linguistics. Willi Hennig, a German biologist who is considered the founder of phylogenetic systematics, in 1950s proposed a method of reconstructing phylogenies that revolutionized the view of the natural order of living beings, which is a product of evolutionary processes. His phylogenetic systematics is based on the detection of exclusive features shared by two sister groups but absent in the third outgroup, the synapomorphies. Let's look at the problem in the graphical way. For simplicity, let's consider just three species: humans, bats, and bees. So here we have three groups: humans, bats, and bees. How to decide which of these three species are more related to each other? So we can look, for example, at the eyes. Humans have eyes, bats have eyes, and bees have eyes. So this trait can't help us to separate or regroup these three species together. So we have to look at the specific traits which differ in these groups. For example, let's look at the number of the extremities, legs and arms. So humans, they have 4, 2 arms, 2 legs. Bats, they have also 4, 2 wings and 2 legs. But bees, they have 6. Okay, so now we have a trait which distinguishes these three groups. Looking at this trait, we might say, okay, let's group together humans and bats, living, bees, and outgroup. So now humans and bats, they form 2 sister groups, and bee is an outgroup. So how we can draw a relatedness from out of here? So we use the lines and we just connect them together. Okay, so they are more related, and then the bees are less related. So now we can connect like this. So this is the plausible diagram if we use the number of extremities as a trait. So let's use something else. Let's use another trait, synapomorphic trait. For example, the genetic systems. So humans have diploid genetic systems, XY. So bats, they also have diploid genetic systems. However, bees, they have haplodiploid genetic systems, which is distinctly different from the genetic systems of bat and the humans. So they have haplodiploid genetic systems in here. So how now we can group? So very logically, they have similarities because of this synapomorphy, and bees form outgroup. So therefore we connect these two together, and then bees will form separate line and we join them somewhere here. So another trait, synapomorphic trait like genetic systems, also produces this type of degrouping and corresponding relatedness. So what if we use some other traits? For example, the ability to fly. So now humans can't fly, bats certainly can, and the bees are excellent flyers. In this case, following this logic of Hennig, so we should group bees and bats together, keeping humans as an outgroup, and then we can connect them together, and only then connect them to the humans. So this apparently wrong phylogeny and the wrong relationship tree in here. So what's wrong here? The ability to flight is not synapomorphic trait. So the ability to fly evolved independently in the bats and in the bees. So it forms this trait as a phenomenon which is called homoplasy. So which means that the trait which has independent origin, but end result is quite similar, the ability to fly. How to resolve this? We have to look at the many traits and then they choose a consensus tree, the tree which would support the relatedness patterns based on the many traits, not only the given one, because given one might be a homoplastic trait, like exactly here. So in particular, how we can resolve this issue here? The type of the flight is quite different between bats and the bees. So the type of the wings and then the frequency of the flapping is different, and dynamics of the flight is different, and different muscles involved, and so on and so forth. So this is homoplastic trait, and therefore it cannot be used to make these inferences. So therefore it's the bottom line. So we have to take many traits and then build a consensus tree which would probably represent the true relationship between species. To understand phylogenetic analysis, let us play a mutation board game proposed by two Brazilian biology teachers. The game simulates the evolution of related individuals along a few generations. We can use it to discuss how series of random mutations in DNA result in phenotypic variation. The aim of the game is to enable us to learn the process of descent with modifications in a playful way. And as well, we can figure out how phylogenetic trees can be reconstructed on the computers using more contemporary current algorithms. Each player has his or her own board showing a phylogenetic tree to be constructed. It presents a segment of the parental DNA that branches into 2 offsprings, then to 4, and then finally to 8 descendant sequences of related individuals. This sequence, generation P, is the same for all players, but the sequences that arise in the following 3 generations, F1 to F3, may be different depending on the history of the copy errors, in other words, mutations. Players use 2 special roulettes to induce mutations. They are printed on the board, and a pencil could work as an axis for a paper when kicked by a finger. The mutation wheel is divided into 6 sectors labeled A, adenine, C, cytosine, G, guanine, and T, thymidine, and plus sign and minus signs. And the 2 sectors labeled with N, no mutations. And the position wheel is divided into equal sectors numbered from 1 to 9, signaling the exact positions in the DNA sequence to be mutated. The goal is to complete the diagram printed on the board by submitting a sequence of DNA bases to successive copying. Error or mutations may occur at each reproduction cycle, at each generation. So now I'm going to play this game on my whiteboard and then demonstrate the use of the mutations in production of diversity. So here is my parental sequence So the sequence contains 9 letters. So this 3, this 3, and this 3. So they form triplets and can be translated to the corresponding amino acids. So these are nucleotides. This is a parental sequence. Instead of the wheels, I'm going to use my computer and application here, which is called wheel of names. Okay, first, so I'm going to spin for the position which I'm going to mutate. So the wheel is spinning and is giving me number 8. So position number 8 is going to be changed. So now I have yet another wheel. So the 8th position is going to be changed for the other letter, which is C. So number 8, 1, 2, 3, 4, 5, 6, 7, 8. So therefore I draw an arrow representing the first descendant, and then just simply say A, C, A, G, A, T, A, and now the letter A is changed to C. I emphasize the C. And then we have the second offspring, so it's going to be F1. So the second offspring. So I have to repeat this process. First I go to the number wheel. Which is giving me position number 9. So what I'm going to have at position number 9. So it's going to be right the same number, the same A. So I am just simply copy it. So it's going to be A, C, A, G, A, T, A, T, A. A is just the same as it used to be. So now I move on to the F2 generation. First the position. Position number 8. So, and it's going to be changed. So, the position C. So this makes the sequence unchanged because we've got C at the position and the new mutation also brings it to C. So I got F2 and then I put right the same sequence A, C, A. So as the first result. So then I have to produce the second offspring in here. But before we do that, let me make a very interesting comment here. So we have one mutation which distinguishes F1 generation sequence from parental sequence at the 8th position right here. So, but what if our next mutation would be back to the T, not to the C as it used to be, but to the T. So then this F2 sequence would be identical to the parental sequence. So this process is called reversal. So, and the reversal usually completely obliterate the direction of the relatedness because it removes the information that the sequence has been propagated over 2 generations. So we've got reversal, then we've got obliteration of the genetic sequence. So if a particular sequence mutates very fast, so there's a high chance or high probability that the reversal mutation will happen. And this will create a significant problem in these methods of reconstruction. So let's try again. So I will return. So I need to generate F2 sequence. So now it's going to be different. Position 7, which is right here. And where I'm going to mutate it. So it says N, no. So it means that this position remains unchanged. So I'm copying this. A, C, A, G, A, T, A, C, A. So now the absence of mutation is another source of the loss of the phylogenetic signal, because now we have two generations with identical sequences. So we would be probably unable to differentiate which one represents generation 1 and which one represents generation 2, because you can see that all the sequences are identical. So the absence of mutation, like what happened right now, results also in the loss of the epigenetic signals and problematic reconstructions here in the sequences. So to prevent that, we have to use, of course, longer sequences where the chance of the same mutation or absence of mutations would be less. Okay, let's look at this part. So I have to go again to my wheel. Okay, position number 4, this G. And I have to mutate it to plus sign. So that is a quite interesting situation. So it means that we've got insertion at that position. So the insertion at that position, it means that instead of one nucleotide and after it, we should add another one, increasing the length of the sequence from the 9 to 10. So let's check what letter I should insert. So it's letter T. So how I go on with this? So I write A, C, A, G, and now letter T, a new letter comes in. This is insertion. A, T, A, T, A. So now I got 10. So this is F2 generation. Now I have to make one more sequence in here. So now this is effect of the insertion. So what if we have thrown minus sign at a given position? That would mean deletion mutation. So for example, let me artificially propagate this with the deletion mutation, and deletion is going to be at the position, let's say 7. So 1, 2, 3, 4, 5, 6, 7, this. And if I throw deletion at the position 7, then what will happen? I will get A, C, A, G, T, 5, 6, and then 7 is eliminated. After that, I will get A, T, A. So that is the effect of deletion, and that one is an effect of insertion. So again, if deletion and insertion happens by chance at the same position, this results into the loss of the phylogenetic signals and perhaps problems in the back reconstructions. Okay, let me fast forward to the full process. So let me show you the results of one of those experiments. So here I mutate the original sequence, parental one, in the 2 offspring F1 generation. So each of these offsprings harbor one new mutation, so substitutions in this case. So when I continue to the F2, we see that offsprings start to harbor 2 mutations in comparison to the original sequence. So here is the deletion appears in the position number 1. So in red, totally here we have 2.
So in the F2 generations, we have 2 mutations. Here is only 1 because I have dialed a non-mutation position in the mutational wheel. So therefore, we have only 1. Then we keep mutating. So here in the F3 generations, of course, we expect 3 mutations, and one of them is deletion. So here is the neutral mutation again. So 1, 2, 3, and again 1, 2, and 3. Here is deletion, new one, and then neutral mutation and T. Okay. So note that the non-mutation is kind of like loss of phylogenetic signal, which might result into the incorrect reconstruction of the tree. The same is true for the deletion. So deletion is the absence of the signal.
These are my sequences produced by the random mutations propagated through the 3 generations in here. So what I normally do in this case, so I collect those sequences in, let's say in this case in PowerPoint, but it can be collected in any desktop application. So here what I do, I use redirect sign and then give a name for the sequence. So here is the parental sequence. This is our original sequence, and these are F3 generation sequences. So sequence 1, 2, and 3. So here I also highlight the mutations, and if I got a deletion event, so then I put a dash sign in here. So what I do afterwards, I basically store this information in the file. So first of all, I copy it, and then I go to the directory, any directory, and then I store this in the file. So it's going to be my game, for example. Yeah? So my board game. If I double click here, and then I can copy the sequence here, save, and now I can rename it. So the extension I can change from txt to fas, and this will enable me to quickly start Mega if it's installed on your computer. So here is the Mega started. Here is the my PAL sequence. Let me zoom in. So now we can explore it a little bit. So here this is the first of all alignment editor, and here we have a parental sequence. So, and then here we have the resultant sequences, and the letters, nucleotides are colored. And here now I can translate it into the protein sequence. It says use the standard genetic code. Yes, we're going to use standard genetic code. And here the nucleotides are translated into the amino acids. And so you can see our parental one is a TDI, and then the sequence variants, let's say the question mark means that the triplet is not complete, but these two are translatable. So we've got a mutant, and here we got tripeptides, and then you can see now they differ from the parental ones, these two, but these 3 are the same, though genetically they are different. So the phenotype, the amino acid sequence, is actually the same. So this table we can use in our quiz, which is the part of the application exercise for this lecture. So then you will analyze how biodiversity is produced through the mutations, both in genotypes, right here, genotypes, and in the phenotypes.